All right. So I've got one more lecture for you about sort of the general principles of molecular visualization before we really start getting into playing with some of these molecular visualization tools a little bit. We'll do that starting on Wednesday. So just a little bit more historical background about some of the software they're going to be looking at. I'm going to talk to you today about how we determine the three-dimensional structure of a molecule using X-rays, and then talk about how a little bit about how that relates to the development of molecular visualization software and kind of where we stand today. So back in the 1800s, we really knew almost nothing about the three-dimensional structure of molecules. We knew which atom was bonded to which other atom, so we could draw a simple stick figure of a molecule, but we really had very little idea about the three-dimensional geometry of that molecule, how those atoms were arranged in three-dimensional space. And this continued right up until the 19 teens. And as you know, as I've mentioned numerous times, atoms are much smaller than the wavelength of visible light. So therefore, you can't use any kind of visible light to actually see what a molecule looks like. But in 1911, a German scientist named Max von Laue discovered that X-rays have a much smaller wavelength than visible light. And he postulated that you could actually use X-rays to determine something about the structure of individual atoms and molecules, because the X-rays were small enough, the wavelength of X-rays was small enough that they could reflect off of atoms, they could be bent by passing between atoms, and you could use that basic principle to then somehow determine something about the structure of molecules. And then it only took a year for someone else to turn that theory into a practical application. So the following year in 1912, William Lawrence Bragg, who was working at one of the research laboratories in London, developed the process of X-ray diffraction using using atoms to basically reflect or diffract, which is really a way of bending a beam of X-rays, using an X-ray beam passing through a crystal to determine the arrangement of atoms within that crystal to determine the three-dimensional arrangement of atoms in a molecule. And he demonstrated this process using an extremely simple molecule initially, which was a crystal of sodium chloride, just basic table salt. One of the challenges with this process of X-ray diffraction, and I'm going to talk a lot more detail about how it works in just a few minutes, but one of the challenges with X-ray diffraction studies, or what's also known frequently as X-ray crystallography, is that it only works if you can get something to crystallize. So what happens in a crystal is that molecules of whatever the substance is are organized in a regular repeating pattern. So that cube of pink and green balls that you see on the left is the crystal structure of sodium chloride, of table salt. And it's simply alternating rows of sodium atoms and chlorine atoms in a very regular defined pattern. And I mean, you've all seen table salt crystals. You know that when you sprinkle salt on your French fries, those are tiny little crystals. You can grow them very slowly and make much bigger crystals of sodium chloride that would have this same repeating pattern throughout. Any other substance that you want to try to analyze using X-ray diffraction also has to crystallize. So that means that even molecules that are much larger than just sodium chloride have to be able to crystallize. So what you're seeing on the right is what it would look like under a microscope if you magnified the sugar that you put on your breakfast cereal. That's sucrose, which is plain old table sugar. Looks like just simple little clear crystals. But one thing you have to realize is that sucrose is a much more complex molecule than table salt. You know, table salt is literally just two atoms, sodium and chlorine. Sucrose is a ring of carbons with an oxygen in it. It's 5 carbons and an oxygen. There's a bunch of hydroxyl groups attached to that. So it's I don't know, I'd have to count it more carefully, but it's 14, 16, 18 atoms, something like that. So that's considerably more complicated than something like sodium chloride, which is just 2 atoms. Now imagine trying to crystallize something like DNA in its double helix path. That's a lot more atoms. Imagine trying to crystallize a protein. Proteins contain thousands and thousands of atoms. And yet many proteins actually do crystallize spontaneously. Some of the first proteins that were studied, and we'll talk more about this later, are things like hemoglobin and myoglobin, which if you just take blood and let it dry out, the hemoglobin in the blood will actually crystallize. If you take muscle tissue and mash it up and allow the liquid from the muscle to dry out, it'll actually form crystals of myoglobin spontaneously. So you can even get some of these large complex molecules to crystallize. Later on in this course, we'll talk about what happens if you have something like a protein that you just can't get it to crystallize. You know, how would you solve its structure? We'll come back to that another time. So the process becomes considerably more complicated as you get progressively more complex molecules, and we'll talk about that more in a minute. So let's just focus initially on something relatively simple like sodium chloride, or a couple of other very simple molecules that have been studied using this same X-ray diffraction technique are things like diamond and graphite. An interesting thing about diamond and graphite, the reason I chose these two examples, is because they have the exact same chemical composition. They're pure carbon. What differs is the arrangement of those carbon atoms. So if you could somehow get a diamond to dissolve and recognize that it's just pure carbon, and if you could do the same thing with graphite, you'd be kind of puzzled. It's like, how is it that the hardest substance, the hardest naturally occurring substance in the world, and one of the softest naturally occurring substances, or at least one of the softest naturally occurring minerals, how is it that they can have the exact same chemical composition? It's because the arrangement of atoms is so different. In a diamond, every carbon is attached to every other carbon. In graphite, you have carbons arranged in thin sheets, and those thin sheets can actually slide relative to one another, which actually makes graphite very slippery. Graphite's an excellent lubricant that's used in a lot of industrial applications. So X-ray crystallography would allow you to pass an X-ray beam through these different crystals and actually determine the 3D structure of the atoms. So how exactly does this work? So here's a simplified view of an apparatus that is used for X-ray diffraction or X-ray crystallography. It consists of a cylinder that's lined with a piece of photographic film. That photographic film is sensitive to any electromagnetic radiation. So if you shine visible light in there, you would expose the film just like you do with a camera. But it's also sensitive to X-rays because X-rays are just a different form of electromagnetic radiation with a different wavelength. So if you shine a beam of X-rays on this photographic film, it's going to expose the film and it's going to create a little black dot on the film. So you do shine a beam of X-rays into the center of this cylinder lined with a piece of film, but in the center of the cylinder, you place a crystal of whatever it is you're trying to study. And that crystal is gradually rotated around 360 degrees inside this cylindrical chamber. And as the X-ray beam strikes the crystal, the X-ray beam is bent at very specific angles, which is a process that we call diffraction, and hits the photographic film in a few very specific spots. And when it hits those spots, it exposes the film. So what you end up with when you do X-ray crystallography, the initial data is just a bunch of black dots on a piece of photographic film. Now here's how it works in a little bit more detail down at the bottom. And I'll admit, there's some of the math that we got to get into here that I don't fully understand, but I understand at least the basic idea of what's happening at the crystal level when we pass an X-ray beam through the crystal. So take a look at this picture down the bottom left. The green dots represent a row of atoms or a layer of atoms that is simply repeating over and over and over again in a repeating pattern. And then there's another layer of green dots right next to that, which is the same atom, again, repeating with that same uniform spacing throughout the entire crystal. when you shine a beam of X-rays at these layers of atoms, they behave as something that physicists would call a diffraction grating. The beam of X-rays is either reflected or bent by these repeated layers of atoms. So X-rays do actually reflect off of atoms, even though visible light doesn't. Now, in this particular situation, we've got the beam of X-rays coming in and hitting the layers of atoms, and I've drawn the X-rays here as a wave, because like all electromagnetic radiation, it travels in a wave form, and the distance between the peaks of the wave is the wavelength of the radiation. So I've drawn these little waveforms coming in and interacting with the atoms on these two layers in this crystal, and then they're bouncing off at some other angle. And if you look really closely at these little waves that are being reflected, you might notice that the peak of one wave lines up with the trough of the wave alongside of it. And then the trough of the first wave lines up with the peak of the wave next to it. This is what we call the waves being out of phase. And believe it or not, when you have waves of energy that are completely out of phase like this, where the peak of one wave lines up with the trough of another, and the trough of the first wave lines up with the peak of the other one, they actually cancel one another out. You can even do this with sound waves. I mean, sound is not a form of electromagnetic radiation, but it is a form of energy that travels in waves. If you set up two speakers on the opposite side of the room, and you have those speakers generate a very specific frequency of sound, or a very specific wavelength of sound, and you offset the two speakers by half the wavelength, and you listen to them, you'll hear nothing. The sound waves, even though both speakers are generating sound, your ear will hear nothing because those sound waves being out of phase are canceling one another out. So what that means is that when the crystal is oriented towards this X-ray beam at certain angles, the X-rays are reflected in such a way that they are out of phase, they cancel one another out, and they don't do anything when they hit the photographic film. They don't expose the film. But as you rotate the crystal, the beam of X-rays is going to strike the layers of atoms in the crystal at a different angle. And so here we see the waves of X-rays hitting the atoms at a different angle. The beam of X-rays then reflects or diffracts off of those atoms in such a way that now the waves are what we call in phase, meaning that the peak of one wave lines up perfectly with the peak of the wave next to it. And the trough of one wave lines up perfectly with the trough of the wave next to it. That means that these waves are actually having an additive effect. The energy of those waves is actually multiplying. And because this is happening throughout the crystal, it's actually going to end up focusing a very strong beam of X-rays on the photographic film. It'll hit the photographic film, and this little black oval here I drew on purpose, that represents the beam of X-rays actually exposing the photographic film and making a black dot on the film. Now, it turns out you can use this to work backwards to actually determine the distance between the layers of atoms in the molecule or in the crystal. And there's a relatively simple mathematical formula. This is actually known as the Bragg equation after William Lawrence Bragg, the guy who discovered it. And I'll explain what all of the terms mean. D is the distance that you're trying to determine, the distance between rows of atoms. So D equals 1/2 lambda, Greek letter lambda, which represents the wavelength. The wavelength is actually the physical distance between peaks of the wave in the beam of X-rays. Lambda over sine of theta. And theta is this angle. the angle at which the X-ray beam is striking the surface of the crystal. Now, there's a principle in physics that the angle of incidence, meaning the angle that the beam is coming into the crystal, is equal to the angle of reflection. So the angle at which these X-rays are bouncing off of the crystal is the same as the angle at which they're entering. So the angle over here on the right would be equal to this angle theta here on the left. So we know what lambda is because the scientist is controlling the wavelength of X-rays that they're generating. And by the early 1900s, we knew enough about X-rays that you could actually control very specific wavelength. And theta is determined by simply measuring the location of that dot on the piece of photographic film. So if you see a dot on the photographic film, you simply measure up from a horizontal axis to where that dot is located, and that tells you this angle theta. So you plug that in, 1/2 lambda over sine theta, and you get d. So you get that distance between atoms in a molecule. So on the left is a photograph of the actual apparatus that William Lawrence Bragg used in 1912 to solve the structure of the sodium chloride crystal. Very primitive looking thing, but it worked. On the right is a much more contemporary X-ray crystallography machine. It still has a cylinder in there, and it'd be shining a beam of X-rays onto a crystal inside the cylinder. Now, it doesn't use a piece of photographic film anymore, just like CTs and X-rays. They now use electronic sensors to sense the beam of X-rays. So similar to the sensors in your phone or your digital camera. And that's actually what these things are up here that are kind of ghosted. And so those are the electronic sensors that are sensing the X-rays. And it kind of looks like instead of rotating the crystal must stay fixed, and instead they rotate the cameras around to detect it. But it still ends up doing the same thing. And essentially what you're doing is just creating a bunch of black dots. Just now you're recording them digitally instead of on a piece of film. So on the left is what one of these X-ray diffraction patterns actually looked like back in the old days when they actually did this with a piece of photographic film. You've got the center of the dot, which would represent, perfectly horizontal. That would represent the X-ray if it just passed straight through. And every single black dot represents one of these reflections, or really diffractions, of the X-ray beam striking the photographic film in phase in such a way that it creates a black dot on the piece of film. And if you look closely, you realize that it's very symmetrical. The left half is identical to the right half. The top half is identical to the bottom half. So you actually don't analyze every single dot. You simply look at one quadrant. You divide it into 4 equal pieces, and you only analyze the dots, say, in the upper right-hand quadrant, because they're going to be identical to the dots in every other part of the pattern. And I think this computer may not have the right font, so it's replaced my font with a Q here. That's actually supposed to say theta, where that little Q is. That's simply measuring the angle, measuring the location of that dot by simply measuring the angle from horizontal. Excuse me. So that gives you your angle theta. You plug that into the Bragg equation, and again, it doesn't have the right font, so it's substituting a lowercase l instead of a lambda here. But d equals 1/2 lambda over sine theta. So d equals 1/2 the wavelength of the X-rays over the sine of that angle. And you come up with a value for d. Well, if you look at something as simple as a sodium chloride atom, what you find is that there are actually many different values for d. D1 up there represents the spacing between rows of sodium atoms, represented by the pink dots, and chlorine atoms, represented by the green dots. So that's one spacing that would actually cause a reflection of the X-ray beam and create a dot on your photographic film. So you'd have some angle theta, and you would solve that and figure out, okay, there's something that's spaced that distance d1 apart. We don't know what it is. We just know there are layers of atoms that are spaced a certain distance, and they're giving us this distance d of d1. And then d2 would be the spacing, say, between one layer of sodium atoms and another layer of sodium atoms. So between the two rows of pink dots, that would be a different value of d, represented by a different dot on the X-ray pattern. D3 could be the distance between chloride or chlorine atoms, again, represented by a different dot on the pattern. But remember that this crystal is also rotated 360 degrees around inside the chamber. So there are going to be other situations where we have these vertical rows of dots that are oriented at the correct angle relative to the X-ray beam. So we're going to get some other distance, d4, the spacing of sodium atoms in a vertical dimension. And then there would be another one, which would be the spacing of the chlorine atoms in a vertical dimension, maybe the spacing between the chlorines and the sodiums in a vertical dimension. But even when the crystal is oriented at some oblique angle, there are going to be repeating rows of dots, repeating rows of atoms in a diagonal fashion. So we have alternating chlorines and sodiums forming a layer, and then another layer of alternating sodiums and chlorines. So what this is pointing out is that even with something as simple as sodium chloride, which is literally just two atoms repeating over and over again, you're going to get an X-ray pattern that actually has multiple dots on it, representing all these different possible distances. So what you're left with is just, you got a sheet of paper. You can do all these calculations by hand because that, d equals 1/2 lambda over sine theta is pretty easy math. You would simply have a list of values of d. You have no idea what they correspond to. But then you would have to sort of by hand and just kind of, you know that salt is sodium ions and chlorine ions. You would then have to try to figure out a three-dimensional geometry that makes sense of those different values of d. So again, imagine how complicated this gets when you put in something even as simple as sucrose, which has maybe 14 atoms in it, because now you got 14 different atoms, even in a single molecule. Now that molecule is repeating itself over and over and over again in the crystal. So you've got all these different atoms that could potentially form flat layers, continuous layers that could be diffracting your X-ray beam. And then imagine that taken even to new levels. You take a protein that has 10,000 atoms in it, and imagine how many different reflections you get, how many different values of d you get. I remember hearing that when the first protein structure was solved, which is 40 or 50 years after this, that when they did the X-ray crystallography pattern from a relatively simple protein, myoglobin, that there were 25,000 different dots on the X-ray pattern that they had to try to analyze. We'll talk more about that in a minute. So here's an example of a molecule that is a little bit more complicated than salt. Actually, it's a fair amount more complicated than sodium chloride. It actually has the name down here, which kind of follows our IUPAC naming conventions a little bit, although part of it you might not recognize. I don't know if you recognize the word pyrimidine, but some of the nucleic acid bases are pyrimidines. It's a simple ring. It's a six-sided ring that includes mostly carbons and one or two nitrogens. And if you look closely at these patterns of dots here, you might be able to tell that there's a ring of something. There's little clusters of black here representing a ring. That would be our pyrimidine ring, which would be carbons and one or two nitrogens. It's 2-chloropyrimidine. So chloro means there's a chlorine ion or a chlorine atom, and 2 tells us that it's on the number 2 carbon. This big atom, these things are almost like topographic maps, like when you're looking at, the topography of land or looking, in geography. So we can tell there's something big up here. That's our chlorine attached to the number 2 carbon of the pyrimidine ring. And then 4,5-diamino tells us that there are two amino groups attached to that same pyrimidine ring. And those are shown over here. This atom is stuck to the ring. That would be a nitrogen with two hydrogens on it. And then there's another one down here. This would be another nitrogen with two hydrogens on it. So those are our two amino groups attached at the number 4 and 5 carbon positions of the pyrimidine ring. So, we got this 6-sided ring with carbons and nitrogens. We've got one chlorine. We got two amino groups with some hydrogens and probably a couple of other hydrogens scattered around in there too. So by biological standards, this is still a very simple molecule compared to something like a protein or compared to something like DNA. And yet, if you look over on the left, what we're looking at is just one quadrant, one corner of the X-ray diffraction pattern. And you see how many dots are produced, how many different reflections of that X-ray beam are produced by even a relatively simple molecule like this. Now, something that's going to become important later in today's lecture, and something I hope you'll remember in the future as you're searching for molecular models, is the following. Especially in the early days when scientists were doing all of this math basically by hand on a piece of paper. If you tried to solve all of the values of D for every single one of those dots in that X-ray diffraction pattern, it would take you, probably wouldn't take you that long to do to solve, D equals 1/2 lambda over sine theta. That wouldn't take you that long. It's not that many dots. But then the process of then trying to reconstruct in 3D what this molecule looks like when all you've got is just a bunch of values of D, that becomes increasingly complicated. I do know, and this is where I start to kind of lose my understanding of this process, is that one thing that these early scientists were doing to take a bunch of values of D and turn that into a 3D model was they were doing something called a Fourier transform, which is a way of taking multiple simultaneous equations and coming up with solutions to them. That's the extent of my knowledge of what a Fourier transform is. I can't do them, and I don't completely understand how it applies to this. But one thing I do know is that, I don't know why my mouse keeps disappearing on me. Let's try there we go. One way to simplify your life a little bit when you're trying to solve the structure of a molecule from an X-ray diffraction pattern is start out by only solving the Bragg equation for the dots that are closest to the center. So that represents small values of the angle theta, right? Where the X-ray beam has been bent less, right? So the beam is closer to horizontal, closer to the center of the piece of film. If you have a small value of theta, and you'll just have to take my word for this, if you plug a small value of theta into the Bragg equation, you end up with a larger value of D, a larger space. And the numbers here on the top of this scale are angstroms. And so what this picture is trying to show, and this small quarter of a circle here, closest to the center of the dot, if you analyze only the dots that fall within that first circle, closest to the center, so that represents small values of the angle theta, but larger values of D, these are all distances that are larger than 2.2 angstroms. And with a computer, you can actually take those values of D, do this thing called the Fourier transform, and actually draw something called an electron density map, which, like I said, it's got, excuse me, it's kind of like a topographic map that's used in geography. In a topographic map, those little lines are measuring elevation. Here in X-ray crystallography, the lines are measuring the concentration of electrons. So if you find lots of lines, that's indicating a cluster of electrons, which tells you there's an atom there. So this first picture is a electron density map that's been drawn by only analyzing the dots closest to the center, which correspond to large values of D. So it says up here, D is greater than 2.2 angstroms. So we're getting a very, very crude picture that there's a molecule here, but we don't really see a lot of detail. Once you've done that, then you can refine the model by gradually analyzing more values of D by studying more of the dots on the X-ray pattern. So after you've studied the dots closest to the center, then you move a little farther out. You analyze some more of the dots that lie a little farther away. These are larger values of theta, but that translates to somewhat smaller values of D. And so this second circle here, if you analyze all the dots that fall inside that second circle, you get a model with a resolution of 1.54 angstroms. And you see that over here, we're starting to get a little bit clearer picture of the molecule. Again, we can tell there's something big up here at the top, and we're starting to get a sense that there's a ring with a couple of things sticking off of it. But these are values or distances, values of T that are greater than 1.54 angstroms. And then you keep doing that. You keep adding more dots to your analysis, keep working your way farther out into the X-ray diffraction pattern. So if you work your way out to this next line and analyze all the dots that fall inside that third line, you get a model with spacings down to 1.19 angstroms, which is actually not that much bigger than the diameter of a hydrogen atom. And so now we definitely get the sense that there's a big atom here in the upper left. You kind of get the sense that there's a ring of atoms down here with a couple more sticking off the side. And then finally, you analyze all of the remaining dots in the pattern all the way out to this outermost line, and we're getting a very, very sharp image, a very high resolution image, all the way down to distances of 0.77 angstroms, which is actually less than the diameter of an individual hydrogen atom. And then you can actually see literally every atom in the molecule on this little topographic map, this electron density map. So this started in 1912 with William Lawrence Bragg solving the structure of sodium chloride. And then the floodgates opened and people all over the world got hold of these machines to do X-ray crystallography. And they just started shoving every crystal they could find into these things and solving the structures of all kinds of solid objects that would form crystals. Jumping ahead 40 years, or 40 years later, X-ray diffraction was the technology that was used to solve the structure of DNA. In this very famous story, I've already recounted some of this for you, but I'll go through the story again with a little bit more detail. So in the early 1950s, there were several groups of scientists around the world who were using X-ray diffraction to try to solve the structured DNA. There were probably more than what we see here, but not a lot more, because you had to be at a very high-tech laboratory to actually have a source of X-rays powerful enough to do this sort of thing. And you needed a lot of very sophisticated equipment. So you had to be at a pretty major university to be doing this. So one of the people who was actively working on this is my buddy Linus Pauling at Caltech, California Institute of Technology, very prestigious school known for their physics and their engineering research. So he had the facilities and the resources to be doing this. The problem was Pauling was not getting great data. His X-ray diffraction images were not very good because he hadn't figured out the trick to getting good X-ray images of the DNA double helix. So based on the crappy data that he was getting, he made a prediction that DNA was actually a triple helix, 3 strands intertwined in a triple coil with the nucleotide bases actually facing outward instead of facing towards one another, towards the interior of the molecule, which from a chemistry point of view made absolutely no sense, but that was the model that was consistent with his bad data that he was getting. Across the ocean, there were a couple of different groups working in England. Watson and Crick, who were the kind of the famous ones, James Watson and Francis Crick, they were working in what's known as the Cavendish Laboratory. It's a physics lab at Cambridge University. Turns out this is the same lab where William Lawrence Bragg invented X-ray crystallography. And they were also getting bad data. They also hadn't figured out the trick to getting good X-ray diffraction images of DNA. So they were aware of Linus Pauling's triple helix model. I think they proposed their own triple helix model at one point in the process, but still weren't getting great results. So meanwhile, outside of London, at King's College, there were two other scientists who were working on this same problem, Rosalind Franklin and her colleague Maurice Wilkins. And Rosalind had found the trick. She knew how to get really good X-ray pictures of DNA. What she discovered, and don't ask me how she figured this out, but somehow she figured out that the experiments had to be performed at very high humidity to maintain hydration of the molecule. You actually had to have water molecules present in the DNA in order for it to maintain its shape. And so she was performing these experiments at very high humidity to keep the DNA hydrated. And she got the best pictures up to that point, the best X-ray diffraction patterns up to that point of DNA. And she shared them with her colleague, Maurice Wilkins. And the two of them both realized right away that, oh, this is clearly a double helix, not a triple helix. And both of them were busy trying to, figure out the details of the structure of DNA. They both had papers in progress, articles, that they were going to publish their findings in progress. And then, without Rosalind Franklin's knowledge, Maurice Wilkins gave a copy of her X-ray data to Watson and Crick. And the story or the rumor is that she did not want to give it to them because she didn't like them. She thought they were cocky and brash and obnoxious and didn't want to share her data with them. But Maurice Wilkins kind of did it behind her back. And they then immediately recognized it's a double helix. They built their famous model out of a whole bunch of mechanical apparatus, figured out that the bases face inward towards one another, and that adenine always matches up with thymine and guanine always matches up with cytosine. They rushed their paper to publication, submitted it to the journal Nature. Rosalind Franklin, when she found out that they were onto something, that they had gotten hold of her data, and that they were rushing their paper to publication, she very quickly finished her paper, submitted it to Nature. I don't know exact timing, a few weeks or a month later. Maurice Wilkins, same thing. He hurriedly finished his paper, submitted it to the journal Nature, also a few weeks, month later, something like that. One of my prized possessions I have at home, I was going to bring it today. I forgot to bring it. Probably shouldn't because it's too valuable. I have a bound set of volume, I think it's 171 from the journal Nature. And I forget what month it was, June maybe, of 1953. All three articles appeared right after another. Watson and Crick's article, followed by Rosalind Franklin's article, followed by Maurice Wilkins' article, literally in order in the same issue of Nature. Watson and Crick got most of the credit because theirs was first, even though the other two made equal contributions. And then in 1962, so 9 years later, the Nobel Prize for Physiology and Medicine was awarded to Watson and Crick and Maurice Wilkins. not to Rosalind Franklin. And everybody always says, oh, this is a terrible oversight, and she didn't get it because she was a woman. But the real reason is she didn't get it because she was dead. She had contracted uterine cancer and had died before the award was announced. And the Nobel Prize Committee never awards Nobel Prizes posthumously. They don't give them to dead. Now, you can argue all day long that that's a really dumb rule, but that's the rule. And there are lots of other people who have been overlooked because they weren't alive anymore. So she was not included in the Nobel Prize. But Watson and Crick, everybody knows Watson and Crick. Nobody knows that Wilkins was also got that same Nobel Prize. But this was all being done with X-ray crystallography. And here's Rosalind Franklin's famous photograph number 51 of the structure of what's called bDNA, which is the naturally occurring form of DNA that would be found in your cells. And I look at that and I see a bunch of dots on a gray circle. Apparently, if you were really into this sort of thing, if you were Linus Pauling or Maurice Wilkins or Rosalind Franklin, you looked at those bunch of dots and said, oh, wow, a double helix. Go figure. Don't know how they know that, but they did. Another little twist to the story is that Linus Pauling was supposed to travel to England earlier in the year and attend a conference in London that would have been attended by Watson and Crick and Franklin and Wilkins. And because Linus Pauling was such a respected chemist, he had already won one Nobel Prize at this point. He was so well respected that people speculate that Rosalind Franklin may have actually shared her data with him willingly. In which case, he may have actually been the first to solve the structure of DNA and rush his paper to publication before anybody else. So he could have been included in that Nobel Prize and won his third Nobel Prize because he had won a second one too at some point. Problem was, he won his second Nobel Prize because he wrote a book called No More War. And it was basically a criticism of war and saying that, wars are destructive and they're going to end the human race. And especially in the early 1950s, with atomic bombs around at that point. And in the 1950s in the United States, if you were anti-war, that must mean you're a communist. So he was blacklisted by Joe McCarthy and the House Un-American Affairs Committee and had his passport revoked so that he couldn't leave the country. So was prevented from traveling to this conference and therefore never had an opportunity to see Rosalind Franklin's data, and so never really got to participate in this discovery. So Watson and Crick made this apparatus. I think I've shown you this picture before, which has we'll talk a lot more about DNA later in the course, but there's what's called a sugar phosphate backbone, which is what holds each strand together. And then you have the nucleotide bases that face in towards the middle. And so they determined that the bases face inward towards one another, and adenine matches up with thymine, and guanine matches up with cytosine in the center of the molecule. So that was their big contribution. And here is their paper from Nature, literally a page. Their whole Nature publication is just literally a one-page article. Then Rosalind Franklin's was like 4 pages, and then Maurice Wilkins' paper was also like 4 pages. But theirs is just literally this one page here with that little spiral diagram in the bottom left showing the double helix. And this is one of my favorite quotes from the history of science. It's just so prescient, meaning predicting the future, but also so understated at the same time, sort of typical British understatement. It has not escaped our notice that the specific pairing we have postulated, in other words, A with T and C with G, immediately suggests a possible copying mechanism for the genetic material. So they predicted that this is how DNA would basically replicate itself by acting as a template, and then attaching the complementary bases to that strand as a way of making copies of the hereditary material. And a lot of people say that Watson and Crick never gave credit to Rosalind Franklin, but you probably can't read it, but she's right there. She gets a credit. They're crediting Maurice Wilkins and Rosalind Franklin from King's College right in their article. So at least they gave her some credit. All right, so the structure of DNA was determined in 1953 by Watson and Crick and Rosalind Franklin and Maurice Wilkins, and published, and was an enormous breakthrough, especially because Watson and Crick pointing out, this copying mechanism for the genetic material. Determining the three-dimensional structure of proteins was quite another matter. Nobody really realized this at the time. They kind of knew what a protein was. They knew that it was a string of amino acids. They could get some proteins to crystallize, like the myoglobin crystals that you see here on the left. But I don't think anybody really had any appreciation for just how complex the internal structure of a protein really is until they started to try to solve them by X-ray crystallography. What you're seeing on the right, it's kind of hard to understand what you're seeing, but the little green squiggles inside that box represent one complete protein molecule, and that's actually a little backbone model of the protein. So all the little squiggles represent all the alpha helices and beta sheets and all those other components of protein structure. And then all the white things represent that same exact protein just repeated over and over and over and over again, thousands of times, tens of thousands of times to create a crystal. So to get a protein to crystallize, you have to literally take the entire protein molecule and get it to stick together in such a way that it repeats over and over and over and over again, thousands of times, tens of thousands of times. And then you shine your X-ray beam into the protein, and you get these tens of thousands of reflections in your X-ray crystallography pattern. Now, there were some earlier attempts to look at some protein structures going all the way back to the 1930s, although they were looking at much simpler proteins that had a relatively simple structure. But there was a guy in the 1930s named William Astbury, and he was working at the University of Leeds in the United Kingdom. And I couldn't tell you exactly where Leeds is in the UK, but apparently it's a place where they have a lot of sheep and they make a lot of wool, and they use that wool to make textiles. So they make a lot of wool fabric and sweaters and suits and tweed jackets and that sort of thing. And so William Astbury actually worked for a textile institute at the University of Leeds in the UK. And because he was interested in textiles, he would take various kinds of fibers and place the fibers inside an X-ray machine and try to conduct X-ray crystallography on the fibers. Now, he didn't really get the fibers to crystallize exactly. He would literally just put a bunch of wool in the machine and blast X-rays, or a bunch of cotton, or a bunch of linen, or he even did things like spider webs and silk. Just anything that he could put inside the X-ray crystallography machine. And he made this interesting discovery that if he took some of these fibers, especially any kind of animal hair, so wool from a sheep, but even human hair, if you put any kind of animal hair into the crystallography device, and you blasted it with X-rays, you get this very, very simple pattern that he called the alpha pattern, which had one dot that corresponded to a distance of 9.5 angstroms, and some other dots that corresponded to a smaller distance of about 5.18 angstroms. And then there were other fibers like silk, I believe, that you could put silk fibers into this, crystallography thing, and you got a different pattern that he called the beta pattern that had dots that corresponded to these other distances that you see listed here. But then the most interesting discovery was if you took one of these fibers that always gave the alpha pattern and you heated it and stretched it, put it under tension, and then subjected it to X-ray crystallography, it would now demonstrate the alpha pattern. Excuse me, the beta pattern, excuse me. So what he speculated was that the amino acid chain was somehow bunched up and gave this alpha pattern. So this would be the typical pattern that you would have for any kind of animal hair made out of keratin. So wool or human hair. And then if you heated it and stretched it, it actually stretched it out into a straight line, which is literally the primary structure of the protein, the linear sequence of amino acids. And so you could convert the alpha pattern to that beta pattern. Whereas there are other proteins like silk that always had the beta pattern, and no matter what you did to them, you couldn't get them to adopt the alpha pattern. So he speculated that at least for these fibrous proteins, like silk and spider web thread and keratin, which made up human hair and wool and cotton and a few other things, that sometimes the strands were bunched up, other times they were nice straight lines, but even the ones that were bunched up, you could heat them and stretch them out into this straight line. But that's about the extent of what he figured out. There's two patterns. Some fibrous proteins are bunched up, some of them are nice straight lines. And you can take the bunched up ones and convert them. I'll give you 3 guesses who solved this problem and figured out what was going on. Who? My favorite chemist of all time? Linus Pauling? Okay, yeah. So what Linus Pauling figured out is that the alpha pattern was actually a spiral of the protein chain, an alpha helix, which we talked about when we were talking about the secondary structure of proteins. Those are the corkscrew spirals that are found in many proteins. And the name alpha helix actually comes from this alpha pattern that William Asprey discovered back in the 1930s. And the beta pattern is literally just the linear strand of amino acids, but they lie parallel to one another and form sheets, which is the beta sheet. And again, the term beta sheet comes from this beta pattern. So here's Linus Pauling next to a space-filling model of the alpha helix that he constructed using these little space-filling models that he invented. So that's the alpha helix, and then here we see, you've seen this picture before, there's the alpha helix. And so when you subjected this to X-ray crystallography, one of the reflections you were getting actually corresponded to the distance between twists of the helix. And then when you heated it and stretched it, you literally were stretching it out into a straight line, and you get a different X-ray pattern, which was actually the repetition of the individual amino acids along the chain. And then the beta sheet were the simple strands of the protein chain lying side by side, connected by hydrogen bonds. And the distance between individual amino acids in a beta sheet is about 3.2 angstroms. So that was responsible for this beta pattern that we saw. Right. So we were starting to talk a little bit about how the structure of proteins was solved, and started out identifying those alpha and beta patterns in the fibrous proteins like keratin and silk and things like that. And then Linus Pauling determined that those represented things like alpha helices and beta sheets. So at this point, we knew about the structure of some of these components of protein structure. These are elements of what we call the secondary structure of a protein. but we still didn't have a sense for the overall structure of a complete three-dimensional protein. That took several years longer. It wasn't until the very end of the 1950s that the very first protein structure was solved. And the reason it took this long is because of something we talked about a little bit before. You've got so many atoms that are repeating themselves over and over again in this crystal that you've got just thousands and thousands of different planes of repeating atoms that are then going to refract or diffract the X-ray beam. And so you've got, in this case, 25,000 different dots on your X-ray pattern, and just an overwhelming amount of data that had to be analyzed in order to start to solve this three-dimensional structure. Now, the folks who finally solved the structure of proteins, they benefited from the fact that the first computers were available. And so they actually used these very primitive computers that were available in the 1950s to crunch a lot of the numbers. Number one, to determine all of those D values, but more importantly, to do all that Fourier transform stuff to start figuring out where the individual atoms were actually located. The picture you're looking at on the left was the very first attempt to illustrate or to model the 3D structure of a protein. Notice the number in the bottom left. It says 6A. That's actually the angstrom symbol, A with a little circle over it. You remember earlier in class, I showed you that one little corner, that one quadrant of the X-ray pattern, and I talked about one way that you can simplify the process of solving the structure of a molecule is to only analyze the dots that are closest to the center first, and then gradually work your way out, analyzing more and more and more dots to gradually get a finer and finer picture of the model. So that thing on the left is the 6 angstrom model of the protein myoglobin. And what that means is that we can't see anything finer. We can't resolve the structure in any more detail than 6 angstroms, which is several times the width of a hydrogen atom. So we're actually not even seeing individual atoms at this point. The guy who was responsible for solving this structure was a guy named John Kendrew, who was also working at the Cavendish lab at Cambridge University, the same lab where William Lawrence Bragg had invented X-ray crystallography in the teens, the same lab where Watson and Crick had determined the structure of DNA. And he created that model on the left based on this very crude, low-resolution data from X-ray crystallography. Turns out every one of those white tubes that you see in the model on the left is actually an alpha helix. So every white tube is actually a complete spiral of the amino acid chain. And you see a more refined picture of it on the right. All those little corkscrew spirals that you're seeing represent the alpha helices that are all coiled up to form that compact blob that you see on the left. Incidentally, this was the structure of sperm whale myoglobin specifically. Sperm whale myoglobin was very abundant and it crystallized very readily. Sperm whales are known for their ability to dive to extreme depths and stay underwater for over an hour, I don't know, maybe even multiple hours at a time on a single breath. And one of the reasons they're able to do that is because they have an enormous amount of myoglobin in their muscle, which temporarily stores oxygen in their muscle tissue. So I think there was still whaling happening in the late 1950s in England. So Kendrew just had to walk down to the local docks and just ask for a big old chunk of sperm whale meat and smush it up and had all the myoglobin he could ever want, and was able to then crystallize it and make a big enough crystal that he was able to do this crystallography stuff on. So after he had determined this kind of low resolution model of myoglobin, he continued to analyze more and more of the dots on the X-ray pattern and gradually refine the model, and eventually was able to create a bunch of those things I showed you earlier called electron density maps that look like these little topographic maps showing the location of atoms, individual atoms within the molecule. And this is a picture of him actually working on the final model of myoglobin in his laboratory. It's a little hard to tell exactly what's going on here, but I'll try to explain what's happening. He had a piece of wood on the floor with a lot of little holes drilled in it. And he had placed little metal rods in all these little holes, creating essentially a forest of little metal rods sticking up. So all those little metal rods you see sticking up are stuck into little holes in a piece of wood on the floor, creating this essentially forest of little rods. And he took those electron density maps that he had created from the X-ray data, and he printed them out on sheets of clear lucite, basically clear plastic. And he put these pieces of clear lucite on their edges, on their sides, within the forest of steel rods, and was able to look on the electron density maps and see where the little circles were, and knew that, okay, there's an atom there. And so he would put a little colored plastic clip onto one of the metal rods at the location where there was an atom that showed up on one of these electron density maps. And he had a whole bunch of electron density maps, creating an entire 3D view of the molecule, and basically just kept putting all these little clips there. And then went through the tedious process of then replacing the clips with little balls to represent atoms and little sticks to represent the bonds between them. And now he did know, we knew enough about proteins at this time to know that it was a string of amino acids. And we had actually solved the linear sequence of amino acids in the sperm whale myoglobin. So he knew that there had to be, an alanine attached to a glycine, attached to a threonine, attached to a proline, whatever. So he knew the amino acid sequence. So he had to attach these atoms together in the right pattern to create that sequence of amino acids, folded up inside this complex blob. And here's what the final thing looked like. Once he had put all the atoms in and removed the forest of steel rods, you have that thing on the left. It's actually not really a ball and stick model. It's just a stick model where the sticks represent the bonds between the atoms. So it's just this cluster of sticks, and to just look at it with the naked eye, it just looks like a complete jumble. But then what he did after that was, again, he knew the amino acid sequence. So he knew that there had to be an amino acid chain in this thing folding itself back and forth to create this three-dimensional blob. So that piece of white string you see is actually essentially a backbone model of the protein, tracing the folding of the amino acid chain throughout this very complex blob. On the right is a painting of that same model by an artist named Irving Geis, who is actually a very important figure in the history of molecular visualization. He was commissioned specifically to paint a picture of that model for the journal Scientific American. Later on, when the second protein structure was solved at the same lab, he was commissioned to do paintings of that molecule as well. In the late 1960s, he actually co-authored a book called The Structure and Function of Proteins with a chemist from UCLA. And the chemist said that I had to make him a co-author because I couldn't afford him as an illustrator. So he actually shared in the royalties on the book. Anyway, it's kind of an interesting guy. I know a little bit about his history. So this was 1958 that John Kendrew finally solved the complete 3D structure of sperm whale myoglobin. So that was the first protein that was ever solved in 3D, down to the level of individual atoms. The painting on the right appeared in Scientific American in 1960, so shortly after that, and took a couple more years before the second protein structure was solved. This is the 3D structure of horse hemoglobin. Apparently, horse hemoglobin was very abundant, very easy to get. You go to your local slaughterhouse where they would actually slaughter horses, actually probably like the glue factory where they would send horses to be rendered into glue. And you just collect all the horse blood, because what else are they going to do with it? And you get the horse blood to crystallize, and then you can study horse hemoglobin. Now, this guy, Max Perutz, was a postdoc who was working also at the Cavendish lab at Cambridge University in England. And he was colleagues with John Kendrew, who had solved the structure of myoglobin. And the story goes that Max Perutz and John Kendrew, they were basically equals. They were sort of equal postdocs. So they were on the sort of the same rung of the hierarchy, the same rung of the ladder. And their boss said, okay, I'm going to assign you two guys to study protein structure. And we've got sperm whale myoglobin from a chunk of sperm whale meat, and we got horse hemoglobin from the local glue factory. Who wants which? They flipped a coin. And Max Perutz got the short end of the deal on this one, because turns out that hemoglobin is exactly 4 times the size of myoglobin. Because hemoglobin is made of 4 separate amino acid chains that are joined together. Each of those chains is almost identical to myoglobin, but it means basically you've got a model that is literally 4 times the size and 4 times the complexity of myoglobin. So it took him 2 more years of crunching numbers with a computer to finally come up with his model. And he came up with this kind of odd looking model on the left with a bunch of white and black slabs. Those did represent the different amino acid chains. Here's another painting by Irving Geis showing that it's basically a bunch of sausages all folded together, but each sausage turns out to be an alpha helix all folded up into this complex blob. And there are 4 separate chains, so 2 dark red ones and then 2 light red ones. And those little discs in there, those represent the heme groups, which are the part of the molecule that actually holds the oxygen atom or oxygen molecule. So by 1960, we had solved the structure of myoglobin and the structure of hemoglobin. So we had the structure of 2 proteins. So even though John Kendrew and Max Perutz in England had worked out the process for determining the 3D structure of a protein, it was so incredibly time-consuming and expensive, and it required so much computing power on these really, really primitive computers that existed at the time, that there were very, very few labs in the world that could replicate that. I think Duke University in North Carolina was one that was able to do some of this work. Linus Pauling got out of this business at this point in his career, so he wasn't continuing to do this at Caltech. I'm not sure where some of the other labs were that were doing this. By 1963, only 3 additional protein structures had been solved. By 1968, only 3 more. So 10 years after Kendrew had solved the first protein structure of myoglobin, we still only had, 3, 6, we only had the structure of 8 proteins in a span of 10 years. So that gives you some idea of how complicated it was and how challenging it was. In spite of that fact, in spite of the fact that by the end of the late 60s, we had only solved the structure of 8 proteins, in 1971 here in the United States, the US government actually launched something called the Protein Data Bank. And it was originally housed at something called the Brookhaven National Laboratories on Long Island, which is really part of the Department of Energy. They do a lot of nuclear energy kind of research. For some reason, they decided to create this protein data bank there. So it was a repository for 3D structural data on the 3D structure of proteins. And when they first opened it, literally had 2 protein structures in it. I haven't checked lately. I think they're up to 500,000, 800,000. It's a lot of 3D protein structures that you can find at this database. So around the same time that these protein structures were being solved, people were starting to use computers to visualize molecular structures. And it started out in a way that was extremely primitive and like everything else, gradually got better over time. This was the first known attempt to use a computer to create a sort of virtual computer model of a protein. This is something called the Kluge system, and I have no idea what that name means or where it comes from. It was developed by a guy named Cyrus Leventhal at MIT. And just to explain what we're looking at here, that little thing that looks like a typewriter in the middle, that's how you entered data into a computer back then, either that or punch cards. but looks like they were typing data directly into the computer on that typing terminal. Parts of the computer in the back, although it probably filled an entire room back in those days. There's a screen over here on the left, and this is long before most computers actually had a screen of any kind. It was actually an oscilloscope, which was a thing that was originally designed for looking at like sine waves, things like that, but it was adapted to display a graphic image of a molecule. And this little clear plastic dome over here, that's an early trackball. So apparently what you would do with this thing is you'd actually have to build a physical model of a protein. So you'd have to build something like that John Kendrew model with the forest of steel rods and all the little sticks. And then you would measure distances and angles and things literally with a ruler and a protractor. And you would enter all of that data into the computer on the little keyboard, the little key punch thing. And you get your model to show up on screen. And then with that primitive trackball thing, that dome, you put your two hands on the dome and you would tilt the dome left, right, forward, and backward, and it would make the model rotate in 3D on screen. But this still required the process of making the physical model first, and then going backwards and sort of measuring the model and putting the data into the computer. The real big revolution in solving the structure of proteins came in the 1970s and 80s, when you could take the X-ray diffraction data directly into the computer. The computer software would then solve the electron density maps and actually build the model right in the computer. So going directly from the X-ray data to the 3D model without ever having to make a physical, hands-on tangible model. And most of that was done throughout the 70s and 80s using a piece of software called Frodo. And I forget where Frodo was developed. I do know that it was designed to run on a very expensive computer system known as an Evans and Sutherland computer system, which is what this guy is working on. And it had a display and it had that really awkward, clunky looking keyboard. The computer system itself was, I don't know, probably $100,000 or something. This is long before IBM PCs or Macintosh or any of that stuff. So it was a very expensive computer system that was really designed and dedicated for this one task. And it ran this application called Frodo. Apparently, J.R.R. Tolkien's estate finally learned about this and objected to the use of the name Frodo. So they simply changed the name to O. So and O still exists. And if you look at those, however many protein models there are in the protein data bank right now, let's say 500,000, probably 400,000 of them were determined using either Frodo or O. Things became a little bit more accessible in the early 80s, or excuse me, early 90s. These machines were fairly expensive, so you had to be like a large university or research lab or something to have one. But molecular visualization finally came to the desktop computer in the early 90s. The very first molecular visualization application that was created to run on a personal computer was an application called Mage. It was released in 1992, only to run on a Mac, and it was produced by Jane and David Richardson. I don't know if you remember her name, but I've talked about her before. She was the woman who basically developed the ribbon representation of proteins, and she was working at Duke University. So she and her husband Dave were one of the reasons why Duke was actually a big center for protein structure at this time. And so, in addition to developing the ribbon representation, she also developed this computer application that allowed you to look at molecular structures on a Mac, on desktop. And she also created a new journal in about 1992, right around the same time. It's a journal called, I think it's just called Structure. It's an entire journal dedicated to understanding molecular structure, especially protein structure. And she included a little floppy disk with a copy of this MAGE software in the envelope with the very first issue of the magazine, of the journal that was released. And so everybody who subscribed to this journal received a copy of this software, and they could start playing around with molecular models on their Mac in their office. The following year, a guy named Roger Sale, who was actually an undergraduate in London, I forget which university he was at, but he developed a tool for also visualizing molecular structure. Originally, he created it to run on Unix-based computers, like on a mainframe computer. But in 1993, he released a version to run specifically on a Mac. It's called RASMOL. And one of the interesting things about RASMOL, a lot of people, first of all, they think it's named after him. His full name was Roger A. Sale, so his initials are RAS. So a lot of people speculated that it was actually his initials in RASMOL, but he says that RASMOL is actually short for raster molecule because it was a computer, way of visualizing molecules on the computer. But not only did he release the RASMOL software itself freely through, but there wasn't really, there sort of was an internet back then, but not as we know it today. He released the source code, which meant anybody could take the actual programming and use it to make other molecular visualization tools. They could incorporate his code into other tools and sort of build off of them. RASMOL was the very first application that I used for molecular visualization, and it was pretty good. I mean, it was primitive, but it did everything I needed it to do. Created pretty pictures, and it even let me export 3D models into my favorite 3D software. 2 years later, there's a company called MDL Information Systems. They took the code for RASMOL, which Roger Sale had freely released into the public domain, and they modified it and embellished it and made a better version called WebLab Viewer Lite. And this was also released just for the Mac in 1995. And this was the second molecular visualization application that I used. So that, this is right when I was really starting to get into doing a lot of molecular illustration work. And I love this application. I wish I could still run this, but I'd have to get like a 30-year-old Mac to try to install it on. So, but again, it did everything I wanted. It created publication quality images. It lets you change between space filling, ball and stick models, ribbon models for proteins. It would export VRML files that you could bring into a 3D program for rendering. It was also around this time that this whole interwebs thing was coming into existence. And so it didn't take long before that same company, MDL Data Systems, to release a version of WebLab Viewer Lite that allowed you to explore and visualize molecules in a web page. So there was this thing called, and I've read the history of this thing 3 different times, and I can never remember whether it's meant to be pronounced chime or chyme. The problem with calling it chyme is that that's what we call like partially digested food that you swallow. But it's supposed to be short for chemical mime. MIME is like something internet mail attachment or something. It's some kind of computer acronym. And so it was meant to have that hard cut sound of chemistry or chemical. So who knows what it's called, but I'm just going to call it chime because that's what it looks like. But anyway, it basically is WebLab Viewer Lite, but it allowed you to embed the molecules into a web page. And you had to have this installed as a plugin in your web browser, but it allowed you to view molecules right in your web browser, rotate them around, change the representation from space filling the ball and stick to ribbon models, etc., and even export the models in various file formats. So jumping ahead to today, there are basically three types of molecular visualization software out there. We're only going to use the first two in here in this class. But there are tools for simply molecular visualization. And they're basically designed to just let you view 3D models of molecules, which you would retrieve from some kind of online database. There are some of these that operate inside a web page. So they're actually, they actually run, in a web page. So there's a list. I won't bother reading the list. It's the one that starts with Karina. So if you go to a database like the Protein Data Bank and you pull up any protein, any molecule, usually you have a choice of 2 or 3 of these different internet tools to actually view the molecule online, right on the database webpage. There are also desktop molecular visualization tools, which are the main ones that I use. That started with that RASMOL thing, and then later with WebLab or Light. But nowadays, we have programs like Chimera, which we've used briefly in here to create our spike model for our coronavirus. VMD, which is short for Visual Molecular Dynamics. It's produced by the University of Illinois. PyMol is produced by a private company. My plan on Wednesday was originally to show you all 3 of these briefly. Every time I try to do this, I find that at least one of them doesn't work. PyMol used to be free. It's now another one that's behind a firewall and you have to pay for it. VMD just stinks. I've never liked it. I show people just because it's another tool you can try, but I'm gonna have to find out today. Maybe I'll check before I leave today and see if these things will even open, because they don't keep them up to date, so they're not necessarily compatible with the newer operating systems. I know Chimera works at least, so at very least on Wednesday we can play around with Chimera a little bit. There are also some molecular visualization apps that function as plugins inside of a 3D program. So there's something called EPMV, the Embedded Python Molecular Viewer, that you can install inside of Cinema 4D. You can install it inside of Blender, 3D Studio Max. I don't think there's a Maya version of EPMV, but that's another one that unfortunately the developers haven't kept up with it. And I can't blame them because it was free. It was great because it was free, but it means that the developers didn't really have any financial incentive to keep up with it. So that may not work so well. There's a tool called Molecular Maya that, as the name suggests, works inside of Maya alone. And then there's a newer one called Molecular Nodes that works inside of Blender. And I know Winnie has some experience with that. It's not one that I've used myself, but Winnie may be able to show us a little bit about how that works. The second type of molecular visualization tool, which we're going to play around with here a little bit in this class, are chemical drawing programs. So let's say you're searching for a model of some small molecule, like some little drug molecule or something like that, and you simply can't find anything in any of the online databases. You can simply draw a little stick figure of the molecule in one of these applications, and it will solve all of the bond angles. First of all, it'll solve the valence. So if you just draw a ring of carbons, it's going to say, well, hey, you're missing a whole bunch of hydrogens. So it's going to fill in the missing hydrogens. And then it's going to determine all the bond angles to create the most energetically stable model of that molecule. And then it'll spit it out as a 3D model for you, which you can then bring into Chimera or into one of these other programs. So we'll play around with at least one of those called Marvin. And then finally, the ones that we're not going to really get into are these computational chemistry programs that do a lot of really high-end stuff that would be mainly of interest just to chemists or structural biologists. They allow you to do a lot of analysis of the molecules about strain and dipole moments and electronegativity and what's hydrophobic and what's hydrophilic and all that kind of stuff. There's also a whole category of these applications that allow you to do genetic sequencing, either sequencing an amino acid chain based on its genetic sequence, or taking either genetic sequences or amino acid sequences from different organisms and determining their phylogeny, their degree of relatedness to them, basically building a family tree from a bunch of molecular data from different organisms. One of the other kind of cool things that some of these applications do is they do what's called ligand docking. So a ligand refers to a small molecule that binds to some bigger molecule. So it could be like the thing that stimulates a protein receptor to do something, like a neurotransmitter. Neurotransmitters are all examples of ligands that bind to various receptor proteins in a synapse. What these programs will do is they will let you just take molecules at random and see if they will bind to a protein. So you've got some, say you've got some receptor protein that does some important thing in the body, and you're trying to discover a drug that's going to stimulate that receptor. Like all of these new weight loss drugs, Wegovy, and I can't remember half the Mounjaro, and I don't know, there's a couple other ones. They're all GLP-1 agonists. So they act kind of like, I think it's like the ghrelin that's released by your stomach that then binds to this GLP-1 protein and says, hey, I'm full. So basically you create an artificial version of that hormone to tell your brain that you're full, even when you're not. And that's how people lose weight on those drugs. So if you know that you've got the receptor that normally binds ghrelin, you've got this GLP-1 receptor, you can just take random models from some database and just plug them into one of these ligand docking applications, and it'll just cycle through them one after another, basically trying to figure out, will this model fit? Nope. Will this model fit? Nope. Will this model fit? Until it finally finds some small molecule that just happens to fit the binding site of that receptor. And that's how a lot of drugs are discovered. All done with what we call in silico. The drugs are actually discovered in a computer by artificially binding, or docking a ligand to its receptor. So, anyway, just a little slideshow here, just a bunch of pictures. These are just screenshots of some of the web-based viewers that allow you to view molecular models right inside a web page, like inside the Protein Data Bank. Here's one of the more common ones that appears inside the Protein Data Bank. Here are some screenshots from some of these desktop applications. So VMD in the upper left, Chimera in the upper right, and PyMol down at the bottom. This is Molecular Maya, just again a screenshot of how you can actually open up a molecule in Maya inside this plugin, and you can do things like change it from a ball and stick model to a surface model to a sphere model to a ribbon model. So you have the ability right inside of Maya to change all different ways of visualizing the molecule. You can play around with different colors and all sorts of things. And then you have all of the regular Maya tools at your disposal. Developed by a guy named Gail McGill, who teaches molecular biology at Harvard, but he also runs a very successful 3D medical animation company called Digizon. Here's EPMV, the embedded Python molecular viewer, developed by Graham Johnson, who's actually trained as a medical illustrator at Hopkins, and he's worked for the Salk Institute, the Scripps Institute, both in California, worked for the Allen Cell Institute for a while. I think he had a faculty position at University of California, San Francisco at some point, but I have no idea what he's up to now. He comes to the AMI meetings all the time, but he ignores me, so I don't know what he does. Here's molecular nodes. There have been a couple of workshops at AMI meetings recently. There was one just this past summer at the meeting we had here in Rochester, so right next door in the Magic Center. I believe it was actually this guy. I believe it was Brady Johnson himself who was demoing the application. So here's a screenshot of this thing called Marvin Sketch. This is one of these chemical drawing programs. So I'll show you this in a couple of weeks. You've basically just got a whole bunch of shapes that you can use to draw bonds, or down here in the bottom left, you've got a bunch of geometric shapes to draw various kinds of rings. So you can draw benzene rings and other kinds of rings. You can draw the bonds in here. Over on the right, you have a bunch of common elements. You can even click, there's a little picture of the periodic table up here. You can click on that and choose any element from the periodic table, and you simply drop those in here and create a very flat looking two-dimensional model. But then the cool part of this is you then hit a button and tell it to solve it in 3D. And that's where it's going to determine the correct bond angles, for the most energetically stable configuration of the molecule. And then you can then export that as a molecular model file that you could then open in any of these other tools that I've just shown you. And final thing I want to talk to you about just very briefly are the different file formats that are used for storing molecular information, molecular model data. The very first file format that was developed specifically for storing 3D molecular structure data was actually the PDB format. So these are files that would literally be .pdb. And that was a format that was developed by the Protein Data Bank when the Protein Data Bank first opened in the early 1970s. I mentioned there was this company called MDL Data Labs that produced the application called WebLab Viewer Lite, which was one of my favorite tools back in the early 90s. They also developed QIIME, which was QIIME, that app that allowed you to view things inside a web page. Anyway, they developed their own proprietary molecular model format called the molecule format. It's literally, the files are .mol. There's a whole bunch of other formats. There's something called MMCIF, which is short for macromolecule crystallographic interchange format. That's another one that was developed by the Protein Data Bank. There's a whole bunch of others. Some of them are specific to particular molecular visualization applications. Some of them were attempts to create sort of universal format. And like most other attempts to make a universal format, it didn't become universal. So it's just yet another format that's out there. Fortunately, most of the databases you encounter are going to have the models in either PDB or MOL format. We do sometimes see the ASN ones. I can't remember a database that actually has MMCIF. ASN one is one that we see occasionally. The good thing is that it kind of doesn't really matter, because most of the modern molecular visualization tools will open any of these formats. So Chimera certainly will open PDB and MOL. It'll open MMCIF, ASN1, probably some of these other formats that are up here. PyMol is the same. In fact, one of the few things that PyMol used to be good for back when it was free was that it would fix bad models. So there were times I would try to bring a model into Chimera and it just wouldn't work for whatever reason. Or I tried to bring it into VMD and it just wouldn't work. I'd open it in PyMol and PyMol would open it fine. Then I'd resave it in a different format out of PyMol and then it would be fine. And I could open it in any program. So PyMol was a great little translator for translating molecules from one format to another, but otherwise not all that great of an application. So on Wednesday this week, I'm going to try right now and see if I can even open VMD and PyMol. I believe they're still installed on these computers, but they may not be compatible with the current operating system. I'll see if I can find that out. And then we'll definitely at least work with Chimera, because I know that one works on Wednesday. All right, so that's everything I have for today.